Hello again. So in this pre-video, we're going to talk about pre-lecture video. We're going to talk about Mendelian genetics. So let me pull up the slides. And we just need to do a slideshow. There we go. So Mendelian genetics is something that you should all be familiar with. And this is genetics that basically was what was discovered by Mendel, which is a fairly uncomplex pattern of inheritance using dominance and recessivity, looking at how traits are inherited. So if we go back to Mendel, so Mendel was somebody we talked about before. This is the monk studying pea plants. And what Mendel did, hide this video panel, there we go. What Mendel did is he basically worked in the 1800s to study how traits were inherited. Now, I realized that he had really no idea what genes were. He had no idea the physical nature. He didn't know he didn't know how genes were inherited. He didn't know that genes were on chromosomes. He didn't know that DNA was the material that encoded the genes. So he had no idea how the physical traits were passed from generation to generation. Mendel comes about the same time that Darwin is in play as well. But Mendel's study was not really that well known and that well understood until the early 1900s. So Mendel and Darwin worked completely separate from one another, came up with some of the same ideas. Darwin looking at populations, Mendel looking at his pea plants. Those ideas were immediately applicable from Mendel to Darwin once they were better understood, but they weren't really understood until the early 1900s. So let's talk a little bit about Mendel. So Mendel basically was a monk living in Austria, and he first started his studies on mice, actually, but mice were not that well tolerated inside of the monastery where he worked, so he switched over to pea plants. He collected data for 10 years. Using basically both self-fertilization and cross-fertilization of the pea plants, and used a lot of statistics to see how different traits were inherited. So what he basically did was he bred the pea plants and then crossed them in so that they had known parents, and observed what the offspring looked like over several generations. So using statistics, he could see how things were inherited. Now, he doesn't really understand what the idea of genes are, so he calls these atoms of inheritance. But the patterns that he understood back in those days really were the same patterns that we understand today. So his conclusions were published in the mid-1800s, 1865, but the scientific world didn't really look at them seriously until the 1900s. So they were almost very obscure until the 1900s. So Mendel has a pea plant. It's a lucky choice because they follow the rules of inheritance very, very well. He had 7 different characteristics, and I think all but one of them, there were 2 that were on the same chromosome but very far apart on that chromosome. All the other ones were on different chromosomes. So when you see inheritance patterns, they are very much inherited independently of one another. Now you can cross these in a directed fashion, so you can determine who the parents will be for each of the generations. And you can look at these observable traits and score them for whether or not they are one phenotype versus another. So the traits that he looked at were round versus wrinkled seeds, yellow versus green seeds, purple flowers versus white flowers, different types of pea pods, so green or yellow pea pods, or inflated or pinched pea pods, axial or terminal flowers, and longer, short stem, longer, short plants, or taller, short plants is the other way that we say this. I think that's actually 8. 1, 2, 3, 4, 5, 6, nope, 7. So 7 different traits, all of which are on different chromosomes, All the genes are on different chromosomes except for 2, and I don't remember which 2. So the way that you perform a cross in pea plants is you basically cut out the anthers before they make the pollen, and then you take the pollen from your desired male and put it onto the stigma of the female. That allows you to do cross fertilization, and if you place a bag over the flower at that point, you prevent other pollen from coming in and fertilizing the plant. You can do self-fertilization by doing the opposite, taking the pollen from the anther, putting it on the stigma, and then of course putting the bag over the plant so it can't get pollen from any place else. So first he started off with what are called monohybrid cross. And every time that we talk about hybrid, hybrid basically means the same as heterozygous. So monohybrid crosses are looking at crosses between 2 heterozygous organisms, and only looking at a single trait. So in order to get his hybrids, first he had to take 2 true breeding plants. And true breeding plants are homozygous. They could be homozygous dominant or homozygous recessive. So when we talk about true breeding, we mean that they are known to be homozygous. And they're known to be homozygous because they've been bred that way for generations after generations. Okay? So the first cross that he did, he took the parental generation, which is the first generation that you look at. He took a tall plant versus a dwarf plant. So he cross-fertilized a true breeding or homozygous dwarf plant with a true breeding or homozygous tall plant. So from that parental generation, so this is the parental generation, you get an F1 generation or a first generation. And what he found was that all of his first generation were uniformly tall. Okay? It didn't matter whether you took the tall plant as the male or the short plant as the male. It didn't really matter which plant was the male and which plant was the female. Your first generation, all of your plants were uniformly tall. Okay? Then he took those hybrids and he did the mono hybrid cross, which is basically taking that first generation and doing a self-cross between members of that generation with each other or with themselves. Okay? Surprise, the F2 generation was not all uniformly tall. The F2 generation had one dwarf plant for every 3 tall plants. So you had a 3-to-1 ratio of tall plants to dwarf plants. And every 4 plants, about 1/4 of them were dwarf, and 3/4 of them were tall. So he figured at that point that there was some sort of a recessive factor, recessive atom of inheritance, that was not able to be seen when there was also the dominant factor in play. So if you mated a tall plant with a short plant, the big T, or the dominant character from the tall plant, basically didn't allow you to see the atom of inheritance that came from the dwarf plant. Okay, but when you did a monohybrid cross, there were some organisms that had 2 atoms of inheritance, or 2 genes, that came from the dwarf plant, and when that happened, you would see that characteristic come out in the phenotype. Okay, and remember, when we talk about this, phenotype is how the organism looks. Genotype is what alleles the organism has. Okay, sometimes we call these a throwback because they go back a couple of generations, but that's a latent factor that's present in all the generations, but you only see it in the phenotype when you see 2 recessive genes coming together. With tallness, if you have 2 dominant genes, 2 dominant alleles coming together, you will see that as a tall plant. But also if you have a tall allele plus a short allele coming together, or dwarf allele coming together, that plant will still look tall. So the phenotypes look different than the genotypes. A heterozygote with the big T, little t genotype will still have the tall phenotype. Okay, so when the F1's reproduced, those recessive genes would recombine in such a way that you could see them if you had two recessive genes coming together. And this, of course, is what it looks like. You've seen this before. Your first generation all has one characteristic, tall or short, or in this case, purple versus white. The second generation will have 3/4 of the dominant characteristic, and 1/4 will be the recessive characteristic. Okay, all monohybrid crosses. So every time that you took two heterozygotes for one trait and crossed them together, you would get a 3-to-1 ratio of phenotypes in the F2 generation. The dominant trait would have the 3, and the recessive trait would have the 1. Okay, so we call these, of course, alleles now. Alleles are alternative forms of the same gene. So we all have those genes. So we all have the gene, for example, for eye color, right? But we have different alleles of that gene, and those different alleles cause us to have different characteristics. So it's not correct to say I have the gene for this. Everybody has that gene, but you may have one certain allele which will give you a certain set of traits. Okay. So this is looking at his crosses. And you can see his numbers here. His numbers are not exactly 3-to-1, but they're really close. So if we did tall versus dwarf, first generation, everything's tall. Second generation, about 3/4 tall, 1/4 dwarf. Okay, you could do your chi-squared on these, and it should come out to be pretty close to being correct. The thing that made this work so well is he did so many, look at all the numbers here. He did so many crosses and counted so many peas and pea pods and pea plants that he could make good statistical analysis. Okay, so terminology, basically the parental generation is the P generation. That's the one that sets up your cross by giving you the idea of what the parents are. The F1 generation are the results of that cross. And then the F1s, when you put them together, when you self them, you'll get an F2 generation. Okay, the monohybrid cross is crossing the F1s together to make the F2 generation. Okay, so when we think about this, and I'm sure you've seen this before, but when we think about this in terms of how do we denote this, usually when you do this, you give a big letter for the dominant trait and a small letter for the recessive trait. Okay, so the parents were big D, a homozygous dominant, and a small d, homozygous recessive. The F1 generation, everybody's a hybrid or a heterozygote, so big D, little d, right? And they're all tall because that big D is dominant to the little d. When you do the cross between those big Ds, or that big D and little d, with another big D and little d, you will get basically out of every 4, 1 will be a homozygous dominant, 2 will be heterozygous, but still display the dominant phenotype, and 1 will be homozygous recessive. Okay? If you take all the guys that are in the F2 generation and realize now you've got 3 different populations, but if you self-fertilize them to get the F3 generation, the next generation, all of these dwarf plants, when you breed them together, will just give you dwarf plants. Okay? Of these tall plants, because we don't know which ones are big D, and big D, little d, you will get some of them that will basically produce all tall plants, right? Some of them will produce tall plants together with the short plants, just like we saw in the F2 generation. Okay? And that depends upon whether you're basically crossing this big D with another big D, another big D, or whether you're crossing this big D little d with another big D little d. Okay, and this is what it looks like in a Punnett square. If you don't remember how to do Punnett squares, the basic idea here is that you need to figure out what the alleles are for both parents. So the allele for one parent goes on the top, the alleles for the other parents go on the side over here, and then you basically put them together in each of the squares of this Punnett square. You can do this for multiple traits at the same time. You don't have to just do it for one trait. And I'll show you a few Punnett squares with that in a little while. But this is the simplest form of how to do these problems. And then what you basically do is you count up the phenotypes and figure out what your ratios are. I will show you another way how to do this in class. It's actually even a little easier than drawing out the square and doing the Punnett square. So Mendel's came up with a bunch of different rules, even though he didn't really understand the genetic basis of what he was seeing. He came up with some really good rules that we still use. The first one is the principle of dominance. And that's basically the idea that you can have dominant genes that will conceal the phenotype of the recessive genes. So if you got a dominant gene together with a recessive gene in a hybrid, the dominant gene is the trait that you'll see. The second one is the principle of segregation, which means that the alleles segregate separately from one another when you make the gametes. Okay, and you can see that in this. If you've got a big D and a little d, the big D will go into one gamete, the little d will go into another gamete. Okay, if you're doing this with multiple different genes, so you're not only looking at the tall versus short, but you're also looking at another separate gene with its allele, you have to realize that that allele segregates separately from the way that the Ds separate, as long as they're on different chromosomes. So the principles of segregation means that the alleles segregate in each generation. They segregate away from each other. And they will do that randomly. And then basically he has what's called the particulate theory of inheritance, which is the idea that genes are inherited unchanged from their parents. Okay, and when you bring those down to your offspring, they are also inherited unchanged. And for the most part, that's true unless there are mutations. Okay, terms that you should know: genotype, phenotype, homozygous, heterozygous. I kind of defined these as we went along. Gametes are the sex cells, so sperm and egg for humans, different ones obviously for different organisms. Punnett square was what we just talked about, and we also defined this one as well. So a dihybrid cross is the second way to do this. So dihybrid cross is looking at two different traits. So in this case, it's looking at P color, P color, yellow and green, okay, where yellow is dominant over green, and round versus wrinkled, where round is dominant over wrinkled. And realize the big R means dominant, the little r means recessive, big Y and little y for the color, okay? So the P generation, the parental generation, what Mendel did is he crossed a true breeding round and yellow plant, so a plant that produced round and yellow seeds, with a plant that produced wrinkled and green seeds, also true breeding. That F1 generation, all of the progeny were round and yellow, okay? And realize this is the Punnett square. It's a really easy Punnett square because you can have a big R coming from this only, and a big Y coming from this only, okay? And a little r and a little y coming from this. This doesn't really distinguish between whether this is coming from the male plant or the female plant, right? But it's a big R and a big Y either case, okay? And then all of the plants that came down from this cross were dihybrid. In other words, they were heterozygous for both the round trait, or both the shape trait and for the color trait. When you do your F2 dihybrid cross, in other words, when you take these plants, the big R, big Y, little y, and cross them together. Do a self-fertilization, or you fertilize them with other brothers and sisters from that same group. What you see basically are all the possible combinations, okay? So realize now when you write out the gametes on this one, you want to make sure that those two traits are represented in each case here, and that you account for all the different gametes that could be produced. So your male plants can give you a big R. You can see the big R here and a big R there. And that big R can be combined with a big Y or with a little y. Okay? You could also get a gamete that has a small y, and that small y could come together with the small r, sorry, and that small r could come together with the small y, or it can come together with the big Y. Okay? So you can see that each of those genes are represented here with the different alleles that can be produced. And then you do the Punnett square, and the Punnett square in this case has 16 squares. And what you will see if you add up all of the possibilities here is that anything that has a big R is round. Okay? So you basically see that you've got all of these round ones here, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, right? So there's 12 round ones to 4 wrinkled ones. And realize that's a 3-to-1 ratio, right? And if you look at yellow versus green, you should see the same 3-to-1 ratio. Okay? But if you look at the two traits together, what you end up with is basically a 9-to-3 to 3-to-1 ratio. You have 9 that are round and yellow. You have 3 that are round and green. You have 3 that are wrinkled and yellow. And you have 1 that is wrinkled and green. And that one is a double recessive. So your lowest number is a double recessive. Your highest number is a double dominant. Okay? And I'll show you in class a different way to get to these numbers, because you can do this easily without necessarily having to do the Punnett squares if you basically just multiply together the numbers of your different gametes to see what you end up with. And I'll show you how to do that a little bit later on. The last rule that Mendel had was the idea of independent assortment. And that rule comes from these dihybrid crosses. And what that means is basically not only do the alleles segregate differently into different gametes, but the genes themselves, the alleles of different genes, different traits, will segregate independently of one another. The big R does not segregate with the big Y all the time. The big R can equally segregate with the big Y, or it can assort with or segregate with the little y. So a gamete could be produced equally well with a big R, big Y, and with the big R, little y. Same is true with the little r, the little y, and the little r and the big Y. And realize that these are in equal proportions. Okay? So that's the end of what we need to talk about in this pre-lecture. So I'm going to save this, and then I'm going to put it up on the site, and hopefully that will give you a little bit of background information before we get into class. So thank you for listening, and I will see you in class.